This morning, it's my pleasure and a privilege to introduce my friend, Dr. Anissa Abi-Dargham. We've known each other, maybe 25 years, and I'm very gratified that she would make the trip here and speak with us today. Anissa is professor of psychiatry and radiology at Columbia University and the New York State Psychiatric Institute. And in a few weeks, she'll become professor of psychiatry and vice chair for research at Stony Brook University. She's recognized internationally as an expert in the area of imaging and psychopharmacology, with a focus on the development of molecular imaging tools to measure neurochemical alterations in the brains of patients with schizophrenia and other neuropsychiatric problems. More recently, she's expanded her research into multimodal imaging in order to examine the functional impact of altered dopaminergic signaling on basic cognitive processes. She's associate editor for the journal Neuropsychopharmacology. She's deputy editor for the journal Biological Psychiatry, past president of the Brain Imaging Council of the Society of Nuclear Medicine, a member of the Board of Scientific Counselors at NIMH, where we run into each other frequently, a scientific advisor for the Schizophrenia Forum, and president-elect of the very prestigious American College of Neuropsychopharmacology. So please join me in welcoming Dr. Anissa Abi-Dargham.
Thank you, Karen, for the nice introduction and also for the invitation. It's been really a special treat to attend parts of this meeting and to visit Geneva. Thank you all for being here on this last day. I hope you can hear me. Is my voice loud enough? Was one? Okay. So what I'm going to do today is present to you our work spanning over the last few decades, looking at dopamine transmission in patients with schizophrenia using PET imaging. And as Karen said, the work really has happened mostly at Columbia University, although my title should include my switch to Stony Brook, which will happen on Friday, July 1st, which is tomorrow. So the outline of my talk, I'm going to first tell you about the hypothesis that was been proposed many years ago about dopamine dysfunction and schizophrenia. Then I'm going to show you the evidence that we have gathered, either to support the hypothesis or in some cases to refine it and to expand it. And then finally, I'm going to conclude with the model that is underlying our current thinking about dopamine dysregulation and schizophrenia. It's commonly thought that dopamine dysregulation is a downstream effect of many other alterations in other neural systems, and that's just the consequences, and when it happens, we have the symptoms. And what I want to bring up today is an alternative model that suggests maybe that dopamine dysfunction is more proximal and could participate in the emergence of dysfunction in other systems even. And it's not just a consequence. So let's talk about the hypothesis and some background on dopamine and just the kind of functions of dopamine that may be very relevant to the dysfunction and the symptom domains we see in schizophrenia. So first, the anatomy. Dopaminergic cells are housed, and this is borrowed from Suzanne Haber's slides. So the dopamine cells are housed in the midbrain and organized in layers. The VTA is in the more ventral layers, and then we have the substantia nigra. They project throughout the brain. From the VTA dopamine cells, those projects to the ventral striatum and to the cortex, while the substantia nigra dopamine cells project to the striatum, the midsection of the striatum, and the posterior section of the striatum. And this is kind of color-coded to show in red the regions that are involved in processing limbic emotional information. In yellow and green are regions that are involved in processing cognitive information. We call these associative regions. And in blue are sensory motor regions. So dopamine is modulating neural systems throughout all these functional domains. Another important property of those dopamine cells is that in the substantia nigra, they mostly fire in tonic mode, which is low frequency. It's under control by tonic GABAergic inhibition and modulated by the potassium and calcium channels as well as the D2 autoreceptor. While VTA dopamine cells have the property of showing burst firing, and this burst firing is driven by excitatory glutamatergic input and inhibition of the GABA input, and it's modulated by the NMDA receptor. This property of VTA dopamine cells, which we've seen project to the ventral striatum and to the cortex, basically gives them the possibility of signaling salience, reward, being able to come on board for cognitive challenges and drive incentive learning. You're probably all familiar, and I've seen the slides many times. This is an example of how dopamine signaling is involved in a reward. You see the increased firing when an unpredicted reward happens in the top panel. In the middle panel, there's no change in firing when the reward is predicted and happens as predicted. While if the reward doesn't happen, despite being signaled by a cue, there's a pause in dopamine cell firing. So this is really at the basis of the positive and negative prediction error signal that is pretty much under control of dopamine that has been essential to studying reward systems in the brain. Another important role of dopamine in modulating behavior is within the striatum, where the medium spiny neurons that project out of the striatum are segregated in two types that bear two types of dopaminergic receptors, the D1 and the D2. The D1 pathway, so medium spiny neurons that have the D1 pathway, form the direct GO pathway, and that is because they project onto the globus pallidum internal and the substantia nigra reticulata. So they inhibit the inhibitory output from these nuclei and facilitate the communication of the thalamus back to the cortex. While the indirect pathway bears D2 receptors, that's the no-go pathway, and have an opposing effect on the communication back from the thalamus to the cortex. And our behavior across all these functional domains, across these cortico-basal ganglia loops, is pretty much under control by this balance and equilibrium between the go, no-go, the D1, D2 pathway. So this is, again, a central role that dopamine plays in organizing behavior and modulating the circuitry. Finally, in the cortex, dopamine projects onto both the GABAergic interneurons and the pyramidal glutamatergic cells and modulates both of them. And this kind of is an intricate interplay of organizing the excitation-inhibition balance and changes throughout development. This is the work of Patricia O'Donnell showing. So there is a maturation of this modulatory role throughout development. So you have a D1 excitation of glutamatergic cell increases between the juvenile and the adult cortex, as you see from the top to the middle panel, while D2 excitation of the GABAergic interneuron exists actually only in the adult brain. So between these two events that happen, you have a kind of a shift from a mode in the prefrontal cortex in the juvenile brain where you have a lot of activity that is not really very well focused to a more focused kind of increased signal to background efficient cortex in the adult brain. And you know, as you see in the lower panel, GABAergic interneuron deficits, which could be either primary or could even be a consequence of altered modulation by dopaminergic receptors, result in an inefficient cortex, which is kind of a similar feature to what is seen in patients with schizophrenia. So I wanted to illustrate with these kind of three nodes of action in the midbrain, reward signaling is the striatum and the cortex, all the roles that dopamine play in modulating critical functions that are abnormal in patients with schizophrenia. So now let's go back to the history a little bit, and why is it that we've been studying dopamine, which actually started in my days with Karen Berman and Danny Weinberger's lab thinking about dopamine and schizophrenia. This has been a kind of accidental discovery with the discovery of chlorpromazine's antipsychotic effects by Delay and Doniker in 1952. Followed by Arvid Carlson's observation that antipsychotics increase dopamine turnover in the brain and led to the formulation of an overstimulation hypothesis of dopamine receptors in schizophrenia by Van Rossum in 1966. Since many discoveries were made, but I will just basically spend some time on this one, that researchers, Jeff Lieberman, Bert Engres, showed that dopamine agonists increase psychosis. So when we put together the fact that dopamine agonists increase psychosis and D2 antagonists lower psychosis, this kind of confirmed the hypothesis of Van Rossum. With the kind of additional detail that is most researchers and the literature was very prominent to refer to this as a mesolimbic dopamine excess. So thinking that the dopamine excess within the striatum should affect mostly the ventral striatum, the limbic striatum. There was a reformulation to introduce the concept of a cortical deficit in addition to the subcortical mesolimbic excess. That happened because of the recognition of the fact that there is a deficit in prefrontal dependent cognition in schizophrenia, which pretty much depends on dopamine. So the idea that there should be an imbalance was put forth. So now what is the evidence? I'm going to take you through this. This is a schematic representation of the dopaminergic synapse in the striatum. You have the presynaptic nerve terminal with the dopamine synthesis machinery, storage, and release. And then you have the postsynaptic medium spiny neuron that here is illustrated with a D2 receptor. You have the postsynaptic and the presynaptic autoreceptor. So the way we look at the dopamine indices in the brain using PET imaging, that we can have radioligand that bind to the D2 receptor and measure D2 receptor density. We can also introduce radioactive DOPA, which basically goes through the synthesis pathway as if it was regular DOPA, becomes F18 dopamine and is taken up by VMAT and stored into the vesicles. So you have a kind of radioactive signal that is correlated to, or it is kind of thought of as a rate of synthesis of dopamine in the brain. Although there are many caveats to all these kind of or mildly confounding factors, but I'm going to ignore those for now. And the third way to look at dopamine transmission is that we can look at dopamine levels. And the way we've done this is basically through the following. We get a baseline scan. Here is in yellow is the radio tracer, D2 radio tracer. It's going to bind in the first, it's hard to point here, we can't point to anything on the slides, but you see the radioactive radio tracer is binding to D2 receptors that are not occupied by dopamine. That gives us a way of measuring the outcome measure, the binding potential. Then we provoke a change in dopamine levels. The middle panel, we have a psychostimulant challenge that produces dopamine release in the synapse. The excess dopamine competes for binding to the D2 receptor with a radio tracer. It also, somehow, excess dopamine will introduce a D2 internalization, increased trafficking. So the bottom line is that the binding potential now will be lower because the radio tracer has fewer D2 receptors to occupy. The change from baseline to post-amphetamine is a correlate of dopamine release, and this has been validated with microdialysis studies. The final panel shows a depletion paradigm. We can remove dopamine from the synapse completely by blocking dopamine synthesis for 48 hours, so basically producing a dopamine-free synapse. Now the radiotracer can bind to all D2 receptors because they're available free of dopamine, and the difference between the baseline scan and the post-depletion scan gives us the amount of dopamine that was in the synapse, basically the occupancy of D2 receptors by dopamine. And this has been used in addition to the ones I've shown you before. I also should tell you that when we combine this with different radio tracers for the D2 receptors that have different levels of affinity, it allows us to image different parts of the brain, D2 receptors in different brain regions. So for example, raclopride allows us to visualize and quantify D2 receptors only in the striatum. Nothing else is reliable. Falipride allows us to image some additional regions that are outside of the striatum, but doesn't have high enough affinity for D2 to visualize the very low levels of D2 in the cortex. For that, we have to use FLB457, which has very high affinity, so we can visualize D2 receptors in the cortex and look at dopamine release this way. Now, despite the fact that FLB binds well in the striatum, we cannot quantify the signal in the striatum. And that is because we do not reach equilibrium in the striatum. So, reaching the point where association equals dissociation, you have kind of a washout of the activity in the striatum. That doesn't happen during the time frame of the experiment. We run out of counts with carbon-11 before we can get to that point, so we cannot quantify that signal. So we get only extrastriatal D2 and dopamine release with FLB. When looking at the striatum as a whole, we have shown that we and others, many labs around the world actually have kind of replicated the finding that there is excess dopamine release in the striatum, excess synthesis, and a very small upregulation in D2 receptors that is mostly present in patients who have had prior antipsychotic treatment. So validating the fact that there's excess D2 stimulation in the striatum. Now, we wanted to test for the mesolimbic hypothesis. We wanted to know whether this excess is mostly in the ventral striatum. For that, we acquired data with higher resolution scanners and were able to do kind of the regional data analysis in collaboration with Suzanne Haber to look at these different functional domains. And what we found was surprising is that although there is excess dopamine, intrasynaptic dopamine, throughout the striatum, as you see in blue, and the blue bars are the patients and the white bars are the controls, and this is the amount of unmasking of D2 receptors after depletion. The biggest unmasking was actually observed in the associative striatum, the midsection, and mostly in the rostral caudate, which was kind of interesting because that's the region that gets the direct projection from the DLPFC, a region of known pathology in schizophrenia, and gets also converging input from limbic region, so it works to integrate across functional domains. So that's where dopamine dysregulation was the highest. We also found that the increase in dopamine release is related to the propensity of amphetamine to increase psychosis transiently in these patients. And this relationship is most exquisitely observed in the associative striatum, the rostral caudate. So the relationship between psychosis and dopamine is mostly about the excess stimulation of D2 in the rostral caudate. Dopamine levels also predicted the response to antipsychotics. This is what we had shown in those patients after they do the imaging study, get treatment. So if they have high levels of dopamine, then the psychosis responds very well when you give them an antipsychotic, which is basically a D2 blocker. And this was confirmed by Kshitish Kapoor's study, where he took patients who are responders non-responders to antipsychotics and healthy controls, and looked at F-DOPA accumulation in the striatum and found that it's increased only in the patients who are responders in that purple bar, while the patients who did not respond in the blue bar had just normal levels of F-DOPA. So psychosis will respond to antipsychotic treatment only if those patients also have excess dopamine. If they have normal dopamine, their psychosis is due to something else than the excess dysregulation of dopamine. They don't respond to our current antipsychotics, which are all dopaminergic. We also looked at the whole brain connectivity of the striatal subdivisions in patients with schizophrenia and controls. You will see here in blue is the rostral caudate. The rostral caudate in controls showed the highest level of connectivity to the rest of the brain. So this region is unique in the sense that it's very connected. This supremacy of the rostral caudate was not present in patients with schizophrenia. So in the rostral caudate, this excess D2 stimulation by dopamine may also affect synaptic plasticity and connectivity of this brain region to the rest of the brain, and this way alter information processing. What about the ventral striatum, and what does dopamine do there in terms of the symptoms of schizophrenia? What we found is that low levels of dopamine there relate to higher levels of negative symptoms. Kind of making sense in a way, since that this is a region where dopamine signaling is crucial to mark, positive, negative prediction errors and to subserve reward functions. That this region, low dopamine, would be related to negative symptoms. We did not find any such relationship to either positive symptoms or negative symptoms in the sensory motor striatum. Now, one thing I didn't tell you about this slide when I showed it to you the first time is that the group, the little graph here where we have dual diagnosis patients, these patients had schizophrenia and addiction. So they're alcoholic or use cocaine, etc. And as a result of the addiction, their dopamine levels are blunted, just as you've seen probably from the lecture of Nora Volkow earlier in the week. These patients share this kind of almost biomarker of addiction, which is blunted dopamine release. Despite blunted dopamine release and levels that are below those in normal controls, these patients still exhibited this relationship between the tiny amounts of changes in dopamine and D2 stimulation and the propensity to increase psychosis. So this, to me, was kind of almost like a moment of puzzlement, because we've just thought that it's the increase in presynaptic dopamine that happens at some point that brings about psychosis. And what this suggested is that there may be just excess sensitivity of the D2 receptor to any stimulation by dopamine that just goes overboard at some point because there's even more excess dopamine release. But there's already something possibly wrong with how D2 receptor processes the interaction with dopamine. So to summarize up until this point, high D2 stimulation in the rostral caudate observes the psychotic symptom domains, while low dopamine in the ventral striatum is related to negative symptoms. I should also mention that this dysregulation and dopamine release, as done by the beautiful work of Oliver House in the UK, was shown to be present before onset of the disease. In the prodromal stage, when subjects come in because they have kind of like non-specific symptoms, but they seem to be almost kind of about to develop a major mental illness and have different outcomes. What he showed is that this group of patients has increased dopamine release in the associative striatum, and that predicts who will convert to schizophrenia. In addition, this excess dopamine progresses, so it increases even further from the index assessment, index scan at first visit, and the onset of the disease. So the dopamine dysregulation is progressing kind of subclinically before people even show the disease. So it's already there. We don't know when does it start. Is it already starting in the premorbid phase? We know it's starting in the prodromal phase. We know it increases more at onset of the disease, and it kind of shows exacerbation during the acute episodes of the illness. But what I want to bring up is that we really don't know how long it's been brewing in the brain before even patients show any symptoms. So in terms of refining the hypothesis, what I've shown you is that it affects mostly the associative striatum, and the dopamine dysregulation precedes the onset of the illness. Now, expanding the hypothesis, looking at the cortex, there's very little data to inform us about the status of dopamine transmission and the frontal cortex in schizophrenia, despite intense interest in this. This is the study of David Lewis showing a decrease in tyrosine hydroxylase immunolabeling in the dorsolateral prefrontal cortex. And we had looked at D1 receptors, we and others, and basically the studies have not been very conclusive and very convergent. And in my opinion, this may reflect the heterogeneity of the illness, may reflect also lack of powers, although some of these studies had large sample sizes. But it also may reflect changes in the D1 receptor with treatment, a downregulation of D1 receptor after antipsychotic medications. So we kind of turned to look at dopamine release as a more direct measure of the availability of dopamine in the cortex. We used FLB457 before and after a psychostimulant challenge. We also obtained working memory assessments and fMRI during working memory performance to look at the correlates of these two. So what you see here is the dorsolateral prefrontal cortex. In blue and red are patients with schizophrenia. Black are the controls. You see the amount of displacement, so how much release of dopamine, how much change, and the binding potential from before to after amphetamine. And you see in black are the controls. They displace on average about 10%, which is in line with the work that was done by Raj Narendran in Pittsburgh to validate this tracer and validate the method and show test-retest reproducibility. So although it's a small signal, it's kind of a reliable signal. In general, controls showed about 10% change. So this is the impact of dopamine release after amphetamine in the cortex. Patients with schizophrenia, on average, showed zero displacement. In addition, both samples as a group showed the same kind of correlation between the amount of dopamine release and the activation on fMRI during working memory performance. So this capacity for dopamine release seemed to be directly related to the capacity to activate the DLPFC during working memory. But this was the most surprising finding. The fact that, as you see, the DLPFC is what we expected to find. You see here in blue the amount of displacement in controls, and in rest, the amount of displacement in patients. And you see all the cortical regions were equally affected: DLPFC, medial prefrontal, orbitofrontal, cingulate, occipital, parietal, temporal. Also, amygdala, hippocampus were affected, thalamus. And most surprisingly, the substantia nigra VTA, midbrain, dopamine cells, where there is some somatodendritic release that can be measured. So throughout the brain, extrastriatal regions all showed this deficit in dopamine release in patients with schizophrenia. This is surprising for many reasons, but I'll get back to that. Let me take you through some of the work of Jared van Snellenberg, a junior faculty in my group, using the self-ordered working memory task that he implemented with fMRI to look at working memory performance in these patients and healthy controls. So let me explain to you a little bit this, actually. So this is a display of these abstract forms, and subjects are supposed to select one shape and kind of remember what they selected, the shapes will reshuffle around the display on the screen, and then they have to select a second shape, which is not the one they selected previously. And since there are 8 shapes, as they go from 1 to 8, it becomes more and more difficult to keep in mind what they've selected previously. So this is really kind of challenging working memory beyond the typical 1, 2, or 3 conditions that are usually used in tasks such as working memory, such as the NBAC task. And using this, what he showed is that there is kind of a decreased performance in both groups as the difficulty of the task increases. But patients with schizophrenia are always performing lower than controls. Here are patients in yellow, which is kind of expected from the literature. We found that there's a correlation between D2 receptor density in the DLPFC and working memory performance. And this was found on the SOT task, on the NBAC, on the letter number sequencing, so it was a very kind of consistent finding. This relationship was present only in healthy controls in red, but not in patients in yellow. And, you know, it's kind of difficult a little bit to understand because one way I think about it is that maybe in healthy controls, if dopamine storage capacity and dopamine levels are normal, the amount of D2 receptor one has will make an impact on their working memory performance. But in patients with schizophrenia, we can detect this because dopamine levels are so low that in a way D2 receptors didn't matter on the performance. The SOT allowed us and Jared to look at test these kind of alternative hypotheses of what's going on in DLPFC in patients with schizophrenia. To explain the deficit in working memory, people have proposed that maybe patients with and the inconsistencies actually in the literature. It's been proposed that maybe patients with schizophrenia have a left shift in their activation to increased difficulty of the task. An alternative hypothesis that they just have disengagement at the higher difficulty conditions of the task. And what Jared has shown is that in healthy controls in black, there's a kind of an inverted U-shaped activation of the DLPFC during performance of this 8-step conditions across the steps. And the more subjects conform to this inverted U-shaped curve of activations versus steps, the better their performance was. Now, patients with schizophrenia, in red, unmedicated, and in blue, medicated, both showed a flattening of this inverted U-shaped curve. In those patients also, the more control-like they were in terms of their inverted U-shape, the better performance they had and better working memory capacity. So this is kind of a neural biomarker linking working memory function to a specific area DLPFC activation during this task, and we hope to try to understand better the biological basis of this inverted U-shaped curve. We found in a subgroup of those unmedicated patients that did the NBAC task a direct relationship between dopamine release and working memory performance. More dopamine release in DLPFC meant better performance. So in a way, although this we didn't find was the SOT, we found it was the NBAC. So what I want to say at this point is that the relationship between dopaminergic indices in the cortex and working memory performance isn't exactly very well defined. And we don't have enough work at this point to have a better understanding. We kind of keep detecting relationships here and there between D2, working memory performance and controls, D1, working memory and patients, as we had shown in the past, dopamine release and working memory performance in some tasks, but not others. So it seems that either there's a lot of heterogeneity and other factors that may come in, or we just don't have enough of a sample to be able to look at all the parameters together and build a model to understand how these impact function. So more to come on this, but we keep detecting something, which just showed us that there is something going on that we just don't have a good grip on yet. So to summarize, prefrontal cortical dopamine is low in patients with schizophrenia and relates to altered cortical function. So expanding the hypothesis, what I've shown you is that there is extrastriatal deficit and most puzzlingly midbrain deficit. And this is, again, the schematic representation. And what this brings about is that if you think about dopamine and the brains of patients with schizophrenia, there is only one discrete area that shows excess dopamine. And that is within the striatum, and in particular the associative striatum. We don't see this excess anywhere else. So that kind of brings up the question as to how this happens. I would have expected that there is excess dopamine cell firing in the midbrain. We would see increased release there because of this excess activity. We don't. So it could be coming from very few fibers that are overactive in the midbrain, projecting to the associative striatum, and that somehow we miss it because they're very few. Or it could be that this is just a terminal event that's happening in the terminal field in the striatum, where regulation of terminal dopamine release is abnormal in schizophrenia, and that's why it's restricted to the striatum and to the associative striatum. So these are the two possibilities that we want to be able to understand and inform. So now let me take you to the model. As I have said, it's easy to think of dopamine as a consequence of other abnormalities, but it could be more proximal than what is generally thought. After all, genetic studies in schizophrenia have shown many loci participating in a small group, small subsets of patients, small proportion, to genetic risk for schizophrenia. But among these loci are features, dopaminergic genes, the D2 gene and others such as AKT, COMT, etc. are affected and are part of the genetic makeup that puts somebody at risk for developing schizophrenia. So we can't ignore the fact that there could be developmental abnormalities in D2 signaling. So what would that mean, and how can we test this? So thankfully, Eric Kandel's lab got interested in these findings of dopamine dysregulation in brains of patients with schizophrenia, and with Christoph Kellendonk and Eleanor Simpson, they engineered a transgenic mouse that has excess D2 stimulation, excess D2 numbers just in the dorsal striatum. D2 were increased by 15%, and that was only during development. So they could turn the gene on and off briefly during development. And so that's the only abnormality this mouse has, only excess D2 in the striatum. And what was profoundly interesting is to see the phenotypes that this mouse exhibited. One of them is that it showed non-reversible deficit in prefrontal dependent cognition. Even when they turn the gene off and D2 goes back to normal, their prefrontal cognition is still impaired. They also have a reversible deficit in incentive motivation, similar to kind of what we call negative symptoms. And so the question was, how is it that D2 signaling in the striatum can affect prefrontal dependent cognition? And there are many mechanisms that were described in this mouse to explain this. What Christoph has found is that there is abnormal dopamine turnover in the cortex. Eleanor Simpson has found that there's decreased firing of VTA dopamine cells. Those are the ones that project to ventral striatum and prefrontal cortex. That is due to decreased expression of NMDA receptors. And Christoph has also found changes in anatomical collateral projections within the basal ganglia. He sees that there are projections from the go D1 to the no-go pathway when the gene is on, and there's D2 upregulation. So, and it has functional consequences. It's like the go pathway is shifted into the no-go pathway, and this can be reversed when the gene is turned off or with chronic haloperidol treatment. So there's all these plastic changes within the brain affecting many off-site targets outside of the striatum, and even affecting the anatomical circuitry within the basal ganglia that obviously would have a big impact on behavior. So I wanted to show this to kind of make the point that abnormal D2 signaling that is kind of like brewing in the brain during development may have consequences on other systems and other brain regions that we haven't really appreciated very much in thinking about schizophrenia. So there's genetic susceptibility factors. Now the environmental susceptibility factors that have been well described in epidemiological studies in schizophrenia suggest that stress, inflammation, drugs, urbanicity, diet, all will have an effect. And what they have in common is that they may all dysregulate dopamine release, increase dopamine activity. So now you have these kind of two events that will kind of synergize and make things worse. So you have the impact on the cells and the molecules and neural circuit function, which gives us the kind of intermediate phenotypes that have been well described: abnormal connectivity, efficiency, synchrony, which may be due to abnormal parvalbumin interneuron functions throughout other brain regions like the hippocampus and the cortex. And then finally, the kind of onset of a massive dysregulation of presynaptic dopamine during adulthood that leads to the clinical syndrome. So this could be one way of thinking about at least some patients with schizophrenia. I'm not saying this applies to all of them. There may be a lot of different biologies for what we think about as the schizophrenias at this point. So finally, the questions and future directions that we are taking. We want to know whether the dual phenotypes, striatal excess and extrastriatal deficit, coexist at a single patient level. We want to be able to image at the same time the midbrain and the striatum and other extrastriatal regions. We want to know what of these abnormalities, the D2 versus presynaptic dopamine, striatal versus extrastriatal, is more primary than the other. And we want to understand whether the global deficit can explain the fact that patients with schizophrenia manifest with so many different symptom domains: psychosis, cognitive alterations, social cognition, executive function abnormalities, etc. And because obviously dopamine is very involved in learning, and abnormal dopamine through development may lead to abnormal learning that will translate into symptoms. So all this has to be kind of understood. And obviously the main conclusion we can derive is even when you think about dopamine only, our drugs, D2 blockers, do not address the kind of level of complexity of the dopamine dysregulation that we've been measuring. One way we're going in the future is to try to understand the cellular mechanisms and the consequences. I mean, we measure these with PET. PET doesn't have resolution to tell us what exact cellular mechanism is occurring in the striatum, for example, that explains what we measure. It could be many things. It could be, for each one of those, possibly more dopamine crossing into the cell, abnormal storage, abnormal function of VMAT, abnormal metabolism, D2 receptors are shifted, etc. I don't want to go through the whole laundry list of possible cellular mechanisms. But one way we're going about this is to work with Jonathan Javich at deriving dopaminergic cells from human-induced pluripotent stem cells obtained from these patients that have extreme phenotypes, extreme excess dopamine release or extreme deficit in dopamine release, and compare those kind of cellular mechanisms of dopamine release, the dopamine machinery, within those dopaminergic neurons and basically in the dish to see what is different between patients and controls that could explain our in vivo results. And with that, we hope to understand better the cellular mechanisms of this dysfunction. So I want to thank my division of translational imaging group, a very special group of very hardworking people. It's hard to point to faces here, but I'll show the names. This is in yellow, the people who've contributed to the work I've shown you, but also collaborators through many years and people at Yale who've helped us with some of the imaging, and my Conti Center collaborators who've contributed a lot to the thinking about dopamine in more mechanistic ways than we could do just imaging. Thank you very much for your attention.